hydrogen ions. So it is 10. It has 10 more hydrogen ions. How does our body resist changes in pH? So our body resists changes in pH through these things called buffers. So buffers are how our body is going to resist them. And then the last question, what body cavity is number 15? So number 15 is pointing down here. It's pointing down to what we know is over here. It's the pelvic cavity. Okay, now let's go to the next one. True or false? A buffer is a solution of weak acid with its conjugate base. And this is true. Next question. The pH scale ranges from 0 to 16. That's actually false. It does not range from 0 to 16 at all. Not even close. That's a very broad range. It actually has a more specific range. It ranges from 0 to 14. So let's go on to the next question. 3. A covalent bond is, this is a positively charged ion where it gives away electrons. So a covalent bond is going to be an ion, that is going to give away electrons, which gives it a positive charge, because at that point it's going to have more protons than it does electrons. So when it gives it away electron, it's going to have more of a positive charge than a negative charge. Next question. Covalent bonds are weak bonds between two partial opposite electrical charges. And this is false. This is not true. That actually is a hydrogen bond that is a weak bond between two partial opposite electrical charges. Next question. An anion is, it's not a positive charge. An anion is when it's negatively charged ion where it gains an electron. So when it gains an electron, it's going to become negatively charged, because now it's going to have more electrons, which are negatively charged, than protons, which is neutrally charged. It's not going to have the same amount, which would make it neutral. So where is hemoglobin? Hemoglobin found? Hemoglobin is found in the blood. It carries oxygen throughout the body. What is the only organic compound that is nonpolar? So the only organic compound that is nonpolar would be lipids. Lipids have they have a characteristic to which they have heads that are hydrophilic and hydrophobic. Their tails are hydrophobic and their heads are hydrophilic. So their heads love water, their heads hate water. True or false? The types of bonds are ionic, covalent, and hydrogen bonds. And that is true. Those are the three main types of chemical bonds. Examples of covalent bonds would be hydrogen and oxygen. Hydrogen and oxygen are examples of covalent bonds because they share electrons. to the next question. True or false? A hydrogen bond, relatively weak, create 3D structures of proteins, enzyme, active sites, and DNA double helix, which is true. They do create 3D structures of proteins, enzyme active sites, and DNA double helix, and they are relatively weak. The strongest of them all would probably be covalent. It takes a lot more energy to break them apart. Hydrolysis reaction are used to break down organic compounds. So let's move on to the next question. What are the 4 types of lipids? The 4 types of lipids are glycerides, sterols, phospholipids, and prostaglandins. Proteins are formed by combinations of how many different amino acids? They're formed from about 20 amino acids. All of them are formed from 20. Respiratory acidosis is a condition that occurs when the lungs can't remove enough of the sugar produced by the body, which is false, because it is when the body cannot remove enough carbon dioxide produced by the body. In water, cohesiveness explains why water beats up. Because water is so attracted to itself. Next question, true or false? Water is a polar covalent bond and a hydrogen bond. This is going to be true. So water is a polar covalent bond and a hydrogen bond. What are all the properties of lipids? So lipids have a lot of properties. They do, like I said, have hydrophilic end, hydrophobic end. They are saturated and they can be unsaturated, and they have like a bar soap kind of texture. They're like slimy. What is the body's primary method of storing fat? So the body's primary method of storing fat is in the form of triglycerides. True or false? Metabolism is the sum of synthesis, anabolism, and decomposition is catabolism, reactions in the synthesis. So this is true. Like I told you in the beginning from our last Kahoot, metabolism is synthesis and decomposition. So it's anabolism and catabolism. Anabolism and catabolism breaking apart and putting together molecules. Let's move on to the next question. So what exactly is pH? So pH is a way of measuring the amount of hydrogen ions that are in a solution. On the contrary, you have OH, which is OH negative. It's another ion. It's how you can imagine how this is how you can neutralize a base. You can identify a base when it has more OH negative than H positive ions. Next question. What is an ionic bond? So an ionic bond is electrically charged ions. So it's when an electron gives away or receives an electron. When an element gives or receives in order to become stable, that creates an ionic bond. Anabolic/anabolism is when you are, so in this case, you are combining 2 components together. You are combining 2 smaller components to make a larger molecule. Okay, next question. A pH of 7 is neutral. That's true. on, next. Decomposition, catabolism is when you are, so this is when you're breaking the molecule apart. You're going to break it apart and create some smaller molecule. Exchange reaction is when you are, so exchange reaction is when there are bonds broken on both sides. So you're going to break bonds on both sides, but you're going to mix it up. It's going to create sort of a new product, but bonds will be broken on both sides and not just one. Okay, so next question. True or false? Synthesis reactions combine reactants into products. So let's see. Synthesis reactions combine reactants into products. That is true. They are like, they're going to combine. They're the sum of metabolism, remember? When reactions require energy, they are considered endergonic. When they require energy, what is the primary storage of carbohydrates in the body? So the primary storage of carbohydrates in the body is going to be glycogen. What are decomposition synthesis reactions used to make? Usually they're used to make organic compounds like sugars, proteins, fats, things of that sort. Let's see. True or false? The 3 stages of replication are initiation, elongation, and termination. That is true. So the 3 stages of replication are initiation, elongation, and termination. You're gonna unzip, copy, and then terminate at a stop. Okay, let's move on. Decomposition reactions. Do they break down compounds into their component? Are exergonic? All of the above. They are exergonic and they break down compounds into all of their components. Into their components. What are the 4 structures of a protein? The 4 structures of a protein are going to be primary, secondary, tertiary, and quaternary. Those are the stages in which the structure of the protein is formed, because it goes through a process in order to shape and become a protein. Okay, let's speed it up. What are enzymes reactions used for? So enzyme reactions are usually used for to lower the activation rate. They're used to make a reaction faster. They can make it more frequent because it lowers the activation energy that is required in order to perform. So it makes it faster and more frequent. Substrates are what we are using to make a product. Enzymes never change, act as glue, product can detach, repeat. This is true. Substrates are like what holds the enzyme together and create the product. Then the product detaches and it can repeat with the enzyme. This is true. Monoglycerides help with digestion and hormones. This is false. They do not do that. That is not their main function. Let's move on. What are the 4 organic compounds? So the 4 organic compounds, earlier I messed up and I accidentally said that lipids were an inorganic compound, but that's not true. Lipids are part of the organic compounds. So it's peptides, nucleic acids, carbohydrates, and lipids. The different components of carbohydrates are, so there's many different components of carbohydrates, and in this case it's going to be all of the above, because monosaccharides, disaccharides, and polysaccharides are all going to be different components of carbohydrates. And monosaccharides, as you can see, is just going to produce energy, glucose and fructose. Disaccharides produce sucrose and lactose, and polysaccharides are going to be multisaccharides. There are other forms of carbohydrates. Nucleic acids. What do nucleic acids do? Transfer energy for the body functions are ATP, DNA, RNA, create new material. They do all of the above. They transfer body energy for body functions. These are examples of them, and they do create genetic materials like DNA and RNA. What is the most common steroid? Is it steroids or cholesterol? I would say it was cholesterol because I mean, you would think it would be steroids, but it's not. It's not. Transmembrane proteins are proteins that are embedded in the membrane. They're proteins that are embedded in the membrane. So in a cell, a cell has transmembrane proteins. Let's move on to channel proteins. Channel proteins, I love channel proteins. They're one of my favorite concepts. They are always going to be, well, usually going to be selective and allow particular material to pass through in and out. So you have sodium and potassium channels. You have all kinds of different channels all throughout your body and within your skin and embedded in your cells, your muscles, all the things. True or false? Passive transport has no energy at all, spread out, move down in concentration. And this is going to be true. They do not have energy, and they do move down in concentration. They spread out. They want to create the concentration and make it even. True or false? Osmosis is the diffusion of sugars. And that is false. Osmosis is just diffusion of water. It's water across the different membranes. Active transport. Let's go. So active transport is the opposite of passive transport. It's going to move against the concentration gradient. So it's gonna instead of having everything be equal, it's gonna want to have all that concentration in one place and not spread out. It's not gonna be equal. Love this. How many sodium molecules and potassium molecules are associated with the NAK bond? And that is, it's going to send 3 sodium out and 2 potassium in every time. It's going to stay in that rhythmic, and like we can get into that later on, but like muscle contractions, sodium and potassium channels is how your muscle is gonna end up to stop contracting in a sense. It's going to get your cell back to homeostasis, your muscle tissue. What is the structure of a carbohydrate? So a carbohydrate is rings of carbon attached to a hydroxyl group. What are the 2 types of endocytosis? So endocytosis is going to be the phagocytes and the pinocytosis. One is cell drinking, one is cell eating. So we have excreting waste. Cytosis is a jelly-like substance that contains the organelles. So this is true. Cytosol is a jelly-like structure that contains the organelles. So let's move on. What is the structure of a glyceride? So the glyceride has fatty acid chains, linear chain of carbons. It has fatty acid chains. Cell differentiation is when the cells become more specialized, and this is true. Cells become more specialized into groups. You have mesoderm, endoderm, and exoderm. Um, all different aspects in which a cell can differentiate. Synthesizes phospholipase, steroid regulates Ca++ concentration. So what does that? That's out of those three, it's going to be the smooth endoplasmic reticulum. It's going to perform these functions. Responsible for sorting, modifying, and shipping off the products that come from the rough ER. So the organ, the part that's going to do this is going to be the Golgi apparatus. It's like the mail sorter. It's gonna sort everything. So this is going to contain ribosome synthesis and modification of proteins for the cell membrane or for export from the cell. So this is going to be the opposite of smooth, and because it contains ribosome, it's going to make it a rough endoplasmic reticulum. Let's move on to number 60. We're almost there. Breaks down and digests unneeded cellular components such as damaged organelle, perform autophagy, or I'm not saying that right, can perform autolysis, and that's going to be the lysosomes. The lysosomes are like the cleanup people. Energy transfer of the cell. That is going to be the mitochondria, aka everybody knows them as the powerhouse of the cell, the energy transformer. When you flip around the equation of cellular respiration, you get photosynthesis. Lipid metabolism and chemical detoxification neutralizes free radicals. So that's going to be the peroxysomes. Because of lipid metabolism and it neutralizes free radicals. That's what gives us away. What is crucial for cell motility, cell reproduction, and transportation of cells within the cell? The cytoskeleton. Cytoskeleton is crucial. Excuse me. Central control center of the cell. This is going to be the nucleus. The nucleus is the control center. It tells everybody what's going on. An envelope surrounding nucleus by lipid bilayer. So it's an envelope. This is going to be the endoplasmic reticulum. It surrounds the nucleus. It's very important. And then you got the smooth and the rough. Transcribing into something new is translation, and that's false. That's not what translation is. True or false? The cell phases are prophase, metaphase, telophase, cytokinesis, and that is true. Mitosis is the replication resulting in two new nuclei, two new somatic cells. Meiosis is cell division that produces it produces gametes. Gametes are sex cells. Almost done True or false? Phospholipids are two fatty acid chains. Phosphorus contain a groove attached to a glycerol backbone with a polar head and a nonpolar tail. That is true. That is exactly what phospholipids are, and that's their structure. Types of cell differentiation.